There's this feeling, and I think we all have it. It's this kind of collective fatigue. It's that moment late autumn you hear about the new flu strain or some new COVID subvariant, and you just realize the booster you got 6 months ago might not cover it.
Right.
It feels like we're constantly on a treadmill, doesn't it? Just sweating, but getting nowhere.
It absolutely does. It's the classic evolutionary arms race.
Yeah.
We are just stuck in this loop.
Yeah.
We develop a vaccine, an antiviral drug,
Mm-hmm.
and the virus, it just evolves a shield. We're always reacting, always one step behind the mutation.
Exactly. We're chasing the burglar after he's already broken into the house. But today we're looking at some research that asks a totally different question.
Yeah.
What if we stopped chasing the burglar? What if, instead of trying to guess which disguise he's going to wear next, we just changed the locks on the doors?
Or, even better, what if we could trick the burglar into walking straight into a trapdoor?
Now, that is a much more satisfying image, and that's really the whole premise of the study we're unpacking today. Mm-hmm.
It was published in Nature Microbiology, and it's titled ARF4-mediated intracellular transport as a broad-spectrum antiviral target.
It's a dense title, I'll grant you that.
Yeah.
But the mission of this deep dive is to, translate that density into something really, really exciting.
Because it's not just about one virus.
No, not at all. This research connects the dots between Zika, influenza, COVID-19, and even vesicular stomatitis virus.
Which is a huge spread. Usually when we talk antivirals, we're talking about very specific targets.
Exactly. So the big idea here is that scientists have found a specific piece of machinery inside our own cells, like a delivery driver, if you will, that all these different viruses hijack. And they figured out a way to fire that driver.
And when you fire the driver, the virus doesn't just get lost.
It gets destroyed. This is a story about turning our own cellular environment against the invader. So let's get into the mechanics of this. To understand the solution, I think we first need to better define the problem with the chase we started with. Why is it so hard to make a drug that just kills these viruses once and for all?
Well, the root of the problem, it really lies in the nature of RNA viruses, like the flu and SARS-CoV-2.
Right.
They have incredibly high mutation rates. They're what we call sloppy copiers. When they replicate their genetic code, they make mistakes constantly.
So our drugs, they're usually targeting the virus itself.
Traditionally, yes. They target viral proteins, things like proteases or polymerases. These are basically the tools the virus uses to replicate.
Okay, so we design a drug that's, like a wedge. It jams into the virus's photocopier so it can't make more copies?
Precisely. That's a perfect way to put it.
Yeah.
But because the virus mutates so quickly, it essentially changes the shape of that photocopier.
So the wedge doesn't fit anymore.
The wedge no longer fits, and that's drug resistance. Suddenly, we're back to square one, needing a new drug, while the virus just keeps spreading.
So the scientists in this study, Lee, and his colleagues, they decided to flip the script. They stopped looking at the virus and started looking at us, the host.
Right. They moved into this realm of host dependency factors.
Yeah.
The concept is actually pretty simple. A virus is an obligate parasite.
Meaning it can't survive on its own?
Not at all. It's just a string of genetic code wrapped in protein. It cannot survive, and it certainly can't replicate without using the machinery inside the human cell. It needs our energy, our materials, our transport systems.
It's like a squatter who moves into your house but needs your kitchen to make a sandwich. So if you lock the fridge, the squatter starves.
That is a great analogy, but there is a massive catch.
Which is?
If you lock the fridge, you might starve too.
That's always the fear with targeting host factors, isn't it? You don't want to kill the patient to kill the virus. If we shut down a process the virus needs, aren't we shutting down a process that we need?
That is the central challenge. The ideal host targets have to be in this Goldilocks zone.
Not too hot, not too cold.
Exactly. It has to be something the virus desperately needs, but that the human body can, technically live without, or, crucially, has a backup for. And that search led these researchers to the ARF family.
ARF, ADP ribosylation factors.
Right.
Sounds like a dog barking, but I'm guessing it's a bit more technical.
A little bit. These are GT passes.
Okay, let's pause on that. GT pace, what does that actually mean?
Think of them as molecular switches. They can be on or they can be off. And when they're on, they bind to a molecule called GTP, that's the energy source, and they help control how vesicles, which are like little cargo pods, move around inside the cell.
So they're the logistics managers. They tell the cargo where to go.
That's it. Now, these ARFs are split into 3 classes.
Huh.
The researchers looked at Class I, specifically a protein called ARF1, and found it is manipulated by viruses. But ARF1 is indispensable.
Indispensable meaning.
What exactly?
Meaning if you mess with ARF1, the cell dies. It's
Ah.
the main power grid. You can't just cut that without crashing the whole system.
Okay, so ARF1 is off limits,
Yeah.
a dead end. But then they looked at class 2, specifically ARF4. And this is where it gets really interesting. They found that ARF4 is the specific accomplice for Zika, for flu, for COVID. It's the specialized delivery driver these viruses really prefer.
But how do we know it's actually helping the virus? I mean, do they just see them hanging out together? Because correlation isn't causation. Maybe ARF4 is just an innocent bystander.
A very fair point. And, scientific rigor demands proof. So they used CRISPR-Cas9.
Or trusty genetic scissors.
Exactly, to completely delete or knock out the ARF4 gene in some cells. Then they exposed those cells to the Zika virus.
And what happened?
The protection was massive. I mean, when ARF4 was gone, viral production dropped by 2 to 4 logs.
We hear logs a lot in science. Can you ground that for us?
Sure. A 1 log reduction is 90%. So 2 logs is 99%. 4 logs is 99.99%.
Wow.
So we are talking about essentially wiping out the virus's ability to replicate.
So without this one protein, the virus is just crippled.
Completely. And to prove it was specific to ARF4, they also knocked out its very close cousin, ARF5.
Which is in the same family.
Same class 2 family. Looks almost identical.
Mm.
Nothing happened. Knocking out ARF5 didn't stop the virus at all.
So the virus has this very specific, unique addiction to ARF4. It's not just grabbing any driver. It needs that driver.
That's exactly it.
Okay, so we've identified the accomplice. ARF4 is the guy. Now, I want to know how the heist works. You said the virus hijacks this driver. Is it just passively using it, or is it more aggressive?
Oh, it's remarkably aggressive.
Yeah.
The study shows that viruses like Zika actively trigger ARF4. When the infection starts, the virus causes the ARF4 that's already inside you to switch into its active state.
It hotwires the car.
It hotwires the car. It turns the key itself. And they proved this with something called a rescue experiment. They took those ARF4 knockout cells, the resistant ones,
Right.
and they added ARF4 back in.
And the infection came roaring back.
Only if they added the normal, wild-type ARF4. If they added a mutant version that was permanently stuck in the off position,
Then what?
the virus couldn't replicate. It needs that switch to flip on in.
Okay, let's try and visualize this, because this is where the mental movie gets good. A virus gets in. It builds its copies in the endoplasmic reticulum, the ER.
The factory floor.
Right. Now it needs to get those copies out of the cell to infect your neighbor.
Correct. And the standard shipping route is from the ER to the Golgi complex.
The Golgi, that's like the shipping dock, the post office where things get packaged and stamped for export.
Precisely. And ARF4 is the truck driver that takes that package from the factory to the shipping dock.
Okay.
Now here's the twist. For me, this was the big aha moment of the paper.
Go on.
When the researchers deleted ARF4, they just expected the virus to get stuck in the factory. Just pile up in the ER.
That would make sense. If the truck doesn't show up, the boxes pile up on the loading dock.
But that's not what happened. The cell didn't just leave the virus there. When ARF4 was missing, the cell actually misrouted the virus.
Misrouted it where?
Instead of going to the Golgi, the viral particles were sent to the lysosome.
Lysosome. Okay, remind us what that does.
The lysosome is the cell's trash compactor. It's the incinerator. It's this little vesicle filled with acidic enzymes designed to chew up waste and recycle materials. It just dissolves everything.
That is incredible. So it's not just that the virus is stuck, it's being actively thrown in the trash.
Yes. They used electron microscopy to actually look inside these cells.
So you can see it happening.
You can see it happening. In normal cells, you see viral particles moving towards the surface. In the ARF4 deficient cells, they saw viral particles trapped inside vesicles that were surrounded by lysosomes, just being degraded.
Talk about a wrong turn. The virus thinks it's heading for the airport to fly to a new host, and instead, it gets dropped into a volcano.
That is a vivid, but an accurate way to put it. By removing the specific driver the virus relies on, the cell's default sorting mechanism takes over.
And it says, what?
It basically says, well, this package doesn't have the right paperwork for the Golgi, so it must be trash.
I love that. It's security by bureaucracy. But here's the problem. We can't just use CRISPR to genetically edit every human to not have ARF4, not today anyway. We need a drug. How do you turn this into a medicine?
That's the crucial transition, right, from basic science to therapy. They knew they couldn't delete the gene in humans, so they looked for a way to block the function of the protein.
To jam the lock.
To jam the lock. And to find that, they looked in, well, in a surprising place, the human eye.
The eye. I saw a reference to rhodopsin. That seems random.
It does, but it comes from previous research. They knew that human rhodopsin, a protein for sensing light, has a little tail that binds to AR4 to help move it around. They realized that interaction site was the key.
So they took that little piece, that tail, and turned it into a peptide.
Correct. They synthesized a peptide they call ARF4-TP4.
Not the catchiest name. We'll call it the blocker.
The blocker works. And its function is really elegant. It acts like gum in the lock. It binds to ARF4 and just prevents it from being activated by the virus.
So the virus jumps in the car, tries to turn the key, but the key won't turn because this peptide is jamming the ignition.
Exactly. And because the ignition won't turn, the ARF4 stays inactive. The truck never starts.
And the virus gets rerouted to the lysosome and destroyed.
That's the mechanism.
So they have this peptide. Does it actually work?
In a petri dish, the results were stellar. Treating cells with the peptide mimicked the genetic knockout almost perfectly. Zika couldn't trigger ARF4, and the virus failed to exit the cell.
But a petri dish isn't a person. It's not even a mouse. Did they test this in animals?
They did. They did extensive mouse studies, starting with Zika. Now, in mice, Zika targets the brain and the testes, causing a lot of damage and inflammation.
Right, which parallels the terrible birth defects we see in humans.
Correct. So they gave mice the peptide and then challenged them with Zika. The results were dramatic. The treated mice had significantly reduced viremia. That's the amount of virus in the blood.
How much of a reduction are we talking?
It was substantial. In the brain and testis, the viral load was nearly eliminated in some of them. But more importantly, they looked at the tissue itself. Usually, a Zika-infected brain shows. Well, it's a mess. Inflammatory cells rushing in, necrosis, cell death.
And in the treated mice?
The organs looked healthy. The structure was preserved.
That is huge. Preventing the physical damage is the whole point. But Zika is one thing. What about something that hits harder and faster, like the flu?
This was the real stress test. They used a lethal dose of H1N1 influenza, a dose meant to kill the mouse.
Okay, so the stakes are high.
Very. In the control group, the untreated mice, only 25% survived.
Oof, that's a brutal mortality rate.
But in the group that was treated with the peptide, survival jumped to 75%.
That is a massive difference. You're tripling the survival rate.
And they didn't just survive, they recovered. The treated mice lost a little weight, which is typical, but by day 14, they had fully recovered. The control mice, well, most didn't make it to day 14.
Okay, I have to play devil's advocate here. We said ARF4 is a host protein. It's part of us. You said targeting host factors is dangerous because you might starve the patient. Did the mice suffer from having their ARF4 blocked?
This is the safety check, and it's absolutely critical. They monitored the mice very closely. They looked at weight, behavior, and specific markers for liver and kidney function, ALT, and creatinine levels.
Yeah.
Everything was normal. No weight loss from the drug itself, no abnormal behavior. Liver and kidneys functioned perfectly.
But how is that possible? You jam the lock on a protein involved in cellular transport, and the mouse is just fine.
It comes back to that idea of redundancy. Biology is resilient. It seems our bodies have a plan B for normal transport. If ARF4 is out of commission, the cell likely uses other ARF proteins, maybe ARF1 or ARF5, to move its own cargo around.
Our cells are like a city with multiple delivery companies. If FedEx is on strike, we use UPS.
Right, but the virus doesn't have a plan B.
It's a specialist.
It's a specialist. It evolved to pick this specific lock. When that lock changes, it's stuck. It doesn't have the flexibility in that moment to switch drivers.
That is incredibly lucky for us. So ARF4 really is that rare Goldilocks target. Vital for the virus, but sparable for the host.
Precisely. It's a narrow window, but they found it.
So we have a mechanism that works for Zika and flu. The paper mentions broad-spectrum potential. Did they test anything else?
They did. They tested SARS-CoV-2, the virus that causes COVID-19 and vesicular stomatitis virus.
And same result.
Same result. Blocking ARF4 suppressed the infection.
This is why we wanted to do this deep dive. We aren't just talking about a cure for the flu here. We're talking about a concept, a broad-spectrum antiviral.
That is the holy grail. I mean, currently, when a new pandemic hits, the timeline is so slow, we have to isolate the virus, sequence it, understand it.
And then design a very specific vaccine or drug that fits that one virus, which takes months, sometimes years.
Time we don't have. But if we have a drug that targets a host pathway, like the ARF4 route that lots of RNA viruses share, the identity of the virus starts to matter less.
It doesn't matter if it's COVID-19 or COVID-24 or some new bird flu.
If it's an RNA virus that uses that delivery driver, this kind of drug could stop it.
That's a game changer. It's like having a master key for the city gates. You just lock the gate, and it doesn't matter what the invading army looks like. They can't get out to spread.
I should offer a word of caution, though. We are looking at a peptide tested in mice.
Right. Manage expectations. I can't go to the pharmacy and buy ARF block tomorrow. What are the hurdles?
Well, peptides are notoriously difficult to turn into drugs. They're fragile. If you took it as a pill, your stomach acid would just digest it like a steak.
So you'd need to inject it?
Probably. Or use nanoparticles to protect it, or modify the chemistry. Turning a peptide into a stable oral medication is a huge engineering hurdle. This study is a proof of concept, a blueprint, but it points the way.
It tells us where to look.
It does. And it validates the entire strategy of host-directed therapy.
Okay, so let's wrap this up. What is the big takeaway for us sitting here, hopefully not fighting off a virus right now?
To summarize, our cells have these bustling shipping routes. And certain viruses, particularly these nasty RNA viruses, have evolved to hijack one specific driver on that route, ARF4.
But we found a way to fire that driver. And when we do that, the cell's own internal security system notices the unauthorized package and sends it straight to the incinerator.
And this worked across multiple different viruses, and it saved lives in animal models without hurting the host.
It's a pretty incredible story of turning the tables.
It really is. You know, we often think of viruses as these master locksmiths constantly picking our locks, outsmarting our defenses, and it feels scary, vulnerable.
It does. It feels like we're always on the defensive.
But this research suggests something else. It suggests that the master thief is actually quite rigid. It relies on very specific habits. And if we just change the locks on the doors we own, if we tweak our own environment just slightly,
the master thief gets stuck inside.
And caught by security.
Yeah.
It raises a provocative thought,
Yeah.
Is the future of medicine not about attacking the invader, but simply about strengthening the fortress? Maybe the best offense really is a good defense.
That is a fascinating thought to leave on. Next time you get a cold, just remember there's a whole factory floor inside your cells fighting a war of logistics. Stay curious, everyone.